So now we come to our final part about droplet microfluidics, and I will talk about our group here at TalTech University. And what I will go through is kind of introduce you first to our group, tell you what we do here, talk a little bit about standard versus high throughput droplet experiments, which is important, something about hetero resistance, and I'll explain more about that, and a little bit antibiotic susceptibility, and just shortly what is the actual kind of principle of our experiments. All right, so this is us. Actually, not all of us, but most of us. So if you at the university you see one of these faces, you can say hi. We are friendly, and we won't scare you away. If you want to know a bit more about us, you can also find us by scanning this QR code. And to kind of sort of give you an overview, we are two parts or two groups within a group. One part of us deals with droplet microfluidics for high throughput assays, while the other group focuses on digital microfluidics for chemical analysis. And also we have a lot of collaborations with other groups and also other universities, and we are a part of the Lab on a Chip initiative here at TalTech. So let's look now at what do we actually do. Well, we mainly work with bacteria. These are single-celled microbes, and they kind of have a genetic information it's contained in one kind of this single loop of DNA. So it looks very much just like a scramble. This is usually the DNA. But some bacteria actually have an extra circle of genetic material, and this is what we called a plasmid. It often contains something that's advantageous for the bacteria. For example, a resistant genes to certain antibiotic. So let's say we have the bacteria, so this is their main DNA, but then they have sometimes these small circles, and on that circle is for example, a resistance gene marker. And what is though then an antibiotic? That is a type of antimicrobial substance which is then active against bacteria. It is the most important type of antibacterial agent for fighting bacterial infections, and the antibiotics are widely used for treatment and also to prevent infection. So antibiotics are really crucial in our society. But then finally, what is antibiotic resistance? Well, that is when actually bacteria become resistant to antibiotics. So if we go over here, we have our sketch that we have our bacteria on a plate here growing, and we want them to be gone. So we put a little bit of antibiotic, we add them. And most bacteria will die from this, but let's say one survives. So when this happens, then the survival will multiply. And then next time when you add the antibiotic, suddenly, because they're all the same genetically, they all suddenly now survive. And this is then what happens when bacteria are resistant to antibiotics. And it's happening a lot in the world now, becoming more and more resistant. So it's a quite serious problem. So as I mentioned, it is a very, and I mean very severe problem, this resistance. But we have then developed Microfluidics tool to investigate this problem, to investigate the antibiotic susceptibility in bacteria. And because we use microfluidics, this enables us high throughput investigation, and it enables heterogeneity in to investigate heterogeneity in antibiotic inhibition at also single cell level. And when we talk about heterogeneity, it means that when you have a bunch of bacteria, as we saw in our previous slide, that is that some get killed, but one single cell of bacteria survives and can multiply and pass on this resistance. So this is what we mean in heterogeneity. And hopefully this knowledge then that we find will contribute in helping scientists and medical doctors in designing more optimal drug treatment in future. But we also have collaborations with other groups at TalTech and other universities. So we help optimizing different droplet microfluidic tools, kind of how to utilize droplet microfluidics for different experiments, not just ours. And also we help with image analysis, and this includes analysis of different substances, particles than droplets. So not only analyzing droplets, but whatever you can kind of throw at us. And finally, big part that we are of the Lab on a Chip initiative. All right, but let's dive in a little bit more about like our standard versus high throughput investigation. So high throughput means just many in parallel, which is very useful with droplet microfluidics. So for us, when you grow usually bacteria and you look at antibiotic resistance, you use usually these plates, and you have your bacteria here growing, and you do experiments. But you need hundreds and hundreds of these stacked piles of petri dishes to kind of, in one experiment, to get your information. While then the high throughput way, what we do with droplets, you can just have a couple of tubes with droplets that each then can have the antibiotic. So hundreds, thousands versus just a couple of tubes. It's very nice to use droplet microfluidics instead, the kind of standard way. But a little bit more now, kind of what actually heteroresistance means, though, because this is very important. Also, why we also use droplet microfluidics. If you look at the definition, it is a form of phenotypic heterogeneity in which seemingly susceptible isogenic bacteria population contain resistant subpopulations. So in simply, this means that bacteria that are genetically identical, they behave differently. So during some antibiotic treatment, some bacteria, they will die. But as we also saw in previous slide, some and it can just be 1 or 2, they still will survive and then multiply and grow further. So in the 1970s studies, where they still used and the plates are still till this day are still also used, they looked at it that if you have this kind of strip here with antibiotic concentration, then you have the susceptible kind of area around it of bacteria. So this is the highest concentration of the antibiotic, and this is the lowest, or 0, basically here. So the higher concentration of antibiotic, you will get this bigger kind of non-growth area. And at a certain point, you can see, okay, you can find the resistance kind of at, okay, from here on until here. Let's say this was like 100. From here, they are actually resistant because they are growing, and they're only very high concentrations can kill them. But in heteroresistance, you can actually see these patches of growth in between. So you have this big area where actually some bacteria die and some still grow at different concentration. So there is no standard limit. Okay, we know from now that they are killed, and below they are not killed. No, here we don't really know, because some grow, some don't. All right. So when we further look at the heteroresistance, we can then see, okay, well, we have the standard experiment where you have one bacteria. Let's say the bacteria is E. coli here. You can have a colony, so right here, and this can really, I mean, millions, billions of cells are actually in that one colony. So it's really hard, you can say, to look at heteroresistance this way, because you have so many cells in one place. And that is also why the droplet microfluidic is very good, because when you encapsulate, you can encapsulate maximum 1 cell per droplet. So this means you can actually look at and study single cell heteroresistance. So in the droplets that actually have bacteria, there's one cell that kind of grows from that, and you can see. But okay, then we can encapsulate these bacteria, but let's then see how we actually study them then. Well, in this study in 2020, Scheller developed a droplet-based digital minimal inhibitory concentration screen. So basically it means that a way to quantify single cell distribution of phenotypic response to antibiotics. It means kind of look at the bacteria single cell level and how they react to antibiotic and how it can vary. So in this system, if we go, also kind of reminds us of the binary system of having just 1 or 0, meaning positive or negative droplet. Then the positive droplets that actually have the bacteria are all scored 1. And if you look at the actual fluorescence intensity, it's very, very high, while kind of the background fluorescence, the empty droplets, are going to be here. So you have a certain number of droplets just being empty, and some with bacteria. And you can kind of then create this threshold and separate these two. So when you add an antibiotic concentration to this, you will then have different, you have a lower kind of number of positive droplets because you have less growth. So you can, let's say, if you study and you add a antibiotic called cefotaxime on the x-axis, so you have a higher concentration, and then you look at how many droplets are positive, or how many bacteria are alive and growing, then you have this nice kind of downward trend, and you see that it's not a straight growing straight line down at a certain point, where there's no growth. You can actually look at heteroresistance. Oh, okay, a lot of, let's say this point, okay, at this concentration, there's still some actually that are growing. Some died, bacteria, but some are growing. And still here, still a couple of resistance. So you can actually find this trend. Okay, there is a difference, and you can study that with different antibiotics. All right, but finally, how does this kind of experiment actually look like? So we will take a look at that. Well, in the beginning, as I said, when we encapsulate one cell of this bacterium. But this still isn't enough to emit fluorescence. So that is also why we then incubate. And for E. coli, it's 24 hours. But if there was a different bacteria, maybe you would have a different time period. And after incubation, though, then we can see if our bacteria, if they are well and growing, if they have multiplied, then we see this nice fluorescent signal. If, let's say, we have added a certain amount of antibiotic concentration to this, then some that actually contained in the beginning a bacteria, they are actually dead. So there is no growth and no fluorescence. And some also droplets will be empty. And from that, there's also no growth. So this way we can distinguish then, okay, are bacteria alive? We score them accordingly, 1, or is the droplet empty or the bacteria dead? Means no growth and scored 0. So after we have this incubation period, we have our bacteria here. And then what we do is to kind of analyze our droplets. We actually pipette our droplets from a test tube into a commercial slide. And when we put them here, they distribute nicely. So they create this kind of a monolayer here. And then you can use different microscope. We use a confocal to kind of make a tile scan. So we go and we actually scan these entire regions. And for each part here, you can have about 5,000 to 6,000 of droplets that you can analyze. So one slide is like, it can be 10,000 droplets or so that you can actually analyze at once. So it's a very nice way to do this. So this brings me to the final conclusion that our our group is here at TalTech University, and I explained that we work with mainly antibiotic resistance and why we actually use droplet microfluidics for this versus the standard way to do it. And I explained a little bit about heteroresistance, that some bacteria, although genetically are the same, they can have different ways they respond to antibiotics, so they can still grow or not grow. And also, finally, I talked a little bit about the principle itself of our experiments, that we incubate them and they fluoresce or they don't. And we then analyze the droplets. And the analysis part will be a part of a different lecture. And finally, of course, have to mention that we are very cool people. So please don't hesitate to contact us at any point if you're interested. And that is it. So thank you for listening.